Welcome. So in the last session, I given a brief introduction of this chapter, Principles of Inheritance and Variation. So in this session, I'm going to discuss why Mendel chose the garden pea plant. So what are the reasons, and then how did he become successful in his experiments? That also we'll see. Then later we will discuss what experiments he performed and the results of those experiments also we will discuss. So let's see why Mendel chose garden pea plant for his experiments. So let's see the reasons for this question. So first thing is the garden pea plant always breeds true. Okay, so here the garden pea plant always breeds true. So here, which means it has actually bisexual flowers, and it will exhibit self-pollination. So usually most of the plants, they go for a cross-pollination. Okay. In cross-pollination, there will be more number of variations seen than in self-pollination. So, but in case of a pea plant, okay, it exhibits self-pollination, and also has bisexual flowers. So it has bisexual flowers and exhibits self-pollination. Okay, then next point, it exhibits distinct contrasting characters. So like yesterday I have given one character, the seed color. So where you can see a very clear cut difference in the characters, like yellow color, okay, green color. So by looking at those characters, we can easily identify, okay, the differences among them. So that is what and another feature the Mendel have chosen in garden pea plant. So it exhibits distinct contrasting characters. Later, what are all those contrasting characters in the pea plant, I will discuss in detail. The next one, the plant, okay, shows, the plant shows, the plant shows short lifespan. So, okay, so it will be growing for, okay, few weeks. So it has a short lifespan and high growth rate and high growth rate, right? So here the lifespan should be less. So if he chooses a plant whose lifespans are very high, then it's quite difficult to come to the results. So he has chosen the garden pea plant, okay, that has a short lifespan. Then the plant can be grown easily and crossed easily. Okay, so if you want, we can go for cross pollination, or we can leave it for, okay, self pollination. So however we want, we can do it. Okay, so that's what the plant can be grown easily and can be crossed easily. That's another point. Then the plant produces large number of offspring. It produces large number of offspring. So I use another word called progeny. So progeny or offspring, okay, one and the same. So it produces large number of offspring. So that actually reduces the error. Then next one, the hybrids, whatever he obtained, okay, by this artificial cross pollination were fertile. So the hybrids obtained by artificial cross pollination okay, were fertile, means they are capable of again reproducing. So whereas in some other cases they may be sterile, so as a result they will not be able to undergo reproduction, but in this case the hybrids whatever he obtained, they were fertile. So these are a few reasons why Mendel have chosen garden pea plant for his experiments. Okay, so the next one we'll see how did Mendel became successful in his experiments. So here, how did Mendel became successful in his experiments? Right, so let's see the reasons how Mendel became successful. So here, Mendel studied only one character at a time. So he studied inheritance of, okay, inheritance of only one character at a time. So he studied only one character at a time. So that is one reason for his success. Not so a plant may have so many characters. So studying all those characters at a time can lead to confusion. But he studied only one character at a time. Then he maintained a neat pedigree record. So he maintained a neat pedigree record. So pedigree record which tells us the parents to offspring, and those offspring later becoming parents and giving again rise to offspring, and so generation after generation all that information he kept, okay, very neatly. He recorded all those things very neatly. So that is, he maintained a neat pedigree record. Then he collected enormous data. So he collected lot of data, okay, for drawing statistical significance. So collected enormous data for statistical significance to, okay, find out the statistical significances. So he took lot of data, okay, and then did these statistics, okay. So collected enormous data for, okay, let's say for drawing statistical significance. So the next one, he had sound knowledge on mathematics. So he has sound knowledge on mathematics, okay, and of course, and also on statistics. So has a sound knowledge on mathematics and statistics, and this made him to get accurate results. So made him to get accurate results, right? So then the last one is selecting garden pea plant for his experiments. So these are reasons, okay. So these reasons are important, okay. So why Mendel chose a garden pea plant, and how did he became successful in his experiments, right? So these are some of the reasons for his success in his experiments, right? So now let's see what experiments he has done and the result of them we will see. Mendel's experiments, right? So here we know that Mendel have chosen garden pea plant. Okay, so here the garden pea plant, whose scientific name is Pisum sativum, and its chromosomal number, so 2n is 14. So it has 14 chromosomes here, okay, in diploid condition. And here let's see those 7 contrasting characters of Pisum sativum. So I told you why did Mendel chose garden pea plant. One of the reason is it exhibits contrasting characters, and there are 7 contrasting, so there are 7 contrasting characters, right? So let's see those 7 contrasting characters in pea plant. So here I'll just mention the character, okay, and the alleles. So these alleles will be either dominant or recessive. Okay, so character and coming to the allele, that may be dominant or, okay, so this is dominant allele and this is recessive allele, or I'll mention in that way. So dominant allele and this is recessive allele, right? So let's see the characters. Okay, first the flower color, right? So this flower color have 2 contrasting characters, okay, violet. So some plants they show violet, okay, flowers, and some others they show recessive, that is a white as recessive. So here dominant is violet and recessive is white. So how did we come to know that violet is dominant and white is recessive? That we will understand slowly. Okay, then the next one, flower position. So flower position, so in this case axillary is dominant whereas terminal is recessive. So terminal is recessive. So here axillary, which means you'll see here, so if the flower is seen, okay, on the stem, so this is axillary, and if it is seen on the tip of the stem, so here, so this is the terminal position of a flower, and this is the axillary position of a flower. So here axillary position, okay, of a flower in case of pea plant, so is always dominant. Okay, so then the terminal is recessive. The next one is seed color. Is seed color. So coming to this seed color, the yellow color is dominant whereas green color is recessive. Then the seed shape, coming to seed shape, in this case, okay, the round shape, so round shape is dominant and wrinkled is recessive. So the round will be seen clear like this, whereas the wrinkle will show in this way. Okay, so this is round shape and this one is wrinkle shape. Okay, then the next one, the pod color. So pod color, so pod, okay, is a sheet that covers the seeds in case of pea plant. So the pod color, green is dominant, yellow is recessive. Okay, so here we can see the pod. So here these are the seeds, okay, after opening it. So this is the pod and these are the seeds. So here the pod color, green is dominant and yellow is recessive. And then coming to the pod shape. So in this case, the smooth or inflated, so that is dominant, whereas constricted is recessive. So constricted is recessive. So if you see this pod very plain, okay, in this way, so this is smooth, and if you happen to see, okay, something like this, okay, wrinkle, then I can say it's a constricted. So it's constricted. So actually very much, okay, close to the seeds. So here you will find something like this. So this is constricted. Is a constricted pod. And if you see, okay, in this way, so then this is inflated. So this is inflated pod. Inflated pod. Okay. Then the last contrasting character is stem height. Character is stem height. So coming to this stem height, the tall, okay, is dominant, and dwarf means short, it is recessive. So these are 7 contrasting characters, and you have to remember, very important. So 7 contrasting characters have total 14 alleles, right? So flower color, flower position, seed color, seed shape, pod color, pod shape, and stem height are 7 contrasting characters. So here uh, in the sessions we'll see, mostly we'll take this okay, seed color, seed shape, and stem height. So I'm going to consider these contrasting characters, okay, and see how Mendel got the results. So first one, we shall say about inheritance of one gene. So the first one, let's see, inheritance of one gene. Right. So this I will also call it monohybrid cross. Monohybrid cross. So inheritance of one gene or monohybrid cross. Okay. So here this is cross between, okay, two different plants. So here two different plants doesn't mean two different species. So the members of the same species, right? Cross between two different plants, okay, which are having one character different. So that is with the plants differing with one character. Okay, so differing with one character. Cross between two different plants differing with the one character. So there is only one character different, the remaining all characters are same. Say, so here we have seen, okay, 7 contrasting characters. So out of 14, okay, only one pair will be different, the remaining all will be same. So here to understand this monohybrid cross, I'm going to take the stem height, the character or the trait I'm going to take is, okay, I'm sorry, stem height. So the, okay, I'm going to take the stem height as the trait or character. So now what Mendel have did is, before he started to perform this experiment, he did self-pollination for a number of times to produce a pure, okay, breeds. So here, okay, I'll just mention that. So stem height has two alleles, we know it, so that is tall and another one is dwarf. Okay, so here the tall, I will take it with the letter capital T, and you have to take the small letter of the same alphabet, okay, for a single character. You cannot take a small d here. Okay, so here taking capital T and here small d will become completely wrong. So here the capital and small letters indicate they are two alleles of a character. Okay, so that's the way we have to do it, and it's very important. So why I have taken here only capital T, because after doing the experiments, okay, we came to know that tall, okay, is dominant. So dominant, okay, characters have to be expressed in capital letters and recessive in smalls. Okay, so dwarf, okay, is recessive and dominant is tall, right? So now what Mendel did, he self-crossed, okay, tall pea plants for a number of generations, so that okay, again if he self-crosses the tall plant with again tall pea plant, he should get again all tall pea plants. There must not be any dwarf pea plant. So if it is the case, then the parents are said to be in heterozygous condition. So he wants to make, okay, those genes homozygous. So in homozygous condition, so a pure line making is, okay, forming into homozygous conditions, right? So he made, okay, a pure, okay, tall breeding pea plant and a pure dwarf, okay, pea plant, right? So let's see, he has taken the parents capital T, and of course here this is what I mentioned, homozygous tall pea plant. So homozygous tall pea plant is crossed with the homozygous, okay, dwarf pea plant. So this is what I say pure breeding, okay, plants. So this is a pure breeding tall pea plant and pure breeding dwarf pea plant, and these are parents, and we know parents are diploid. So hence I need to take, okay, two alleles. I need to take two alleles here. So because they are diploid in condition, and for every character there will be two alleles in a diploid organism. So hence I need to take here, okay, two, right? So this is a homozygous tall pea plant. So already we have understood in our earlier session what actually is this homozygous condition, and this right, of course, homozygous, both are homozygous conditions. Heterozygous, which means you need to take capital T, small t. If it is the case, then it is heterozygous condition. Now during the formation of gametes, so meiosis, and it results in the formation of okay, of course, the gametes. So here meiosis and gametes are formed. So these gametes, okay, have to be circled. So we should not circle the parents. Okay, so generally we follow it as a rule, okay, in genetics, that we put the gametes in circle. So here again, out of these two, okay, only one will be present in the gamete, because we know while the formation of gametes, the number of okay, chromosomes will be half to that of the parent. I hope okay, you might have remembered the okay, well, we discussed about cell cycle and division in the process of meiosis. So in the process of meiosis, okay, the chromosomal number will exactly reduce to half to that of its parent. So hence the genes present on them will also reduce exactly to half. So hence, as here I have taken okay 2n okay parents, okay, with the two capitals, so only half of this will be in the gamete. So that's why I have mentioned only one T out of these two. So here also the same condition, right? And I told you only gametes have to be circled, and these are gametes, okay, which are haploid, right? And now, so both together, okay, fertilization, then it results in capital T, small t. So here this is the F1 generation. So here F actually stands for okay filial. So F stands for filial, okay, which means son, right? And usually the generations we will represent as okay, if it is the first generation, then we call it F1 generation, so which means filial 1 generation, because I'm considering from this point. Okay, of course, to these there may be earlier parents, but I'm not considering from that point. So here from this point I'm considering for that reason. So when I have taken these as parents, okay, their offspring will be the first generation, and from this, if I'm going to produce the next generation, then it will become F2 generation, and from them the next will be F3 generation, and it goes on. Okay, so this is starting from this point as I have taken. So here I have mentioned it F1 generation, which means okay, filial 1 generation. Now, okay, when Mendel have taken tall pea plant and a dwarf pea plant, which are homozygous, and when he crossed, okay, and he obtained seeds, so many, say suppose 1,000 seeds, say suppose he obtained 1,000 seeds, and these 1,000 seeds he sown and then again raised. When he raised, okay, all the pea plants were grown okay tall. So the question comes, where did this dwarf character gone? Okay, so Mendel was surprised to know that this dwarf character was not expressed in F1 generation. So it was not expressed in F1 generation. Okay, so when he crossed a pure tall okay breeding pea plant with the dwarf one, so he obtained all tall plants. So all these F1 generation, he obtained all tall pea plants. All are tall pea plants. So I'll just mention it, 100% tall pea plants. So there was not even one single plant that is dwarf. Okay, so right now what he did to find out what happened to that okay dwarf character, so he gone for self crossing. That's okay. So Mendel self-crossed the F1 generation. So Mendel self-crossed the F1 generation. So what is the F1 generation is capital T, small t. So this is what we have obtained. Okay. So but to external, that is the phenotype, all are tall. He don't know whether inside the dwarf character is present or not. So whatever that in the F1 generation, the P plants he obtained, he took two P plants and he crossed them. Okay, so that is Mendel crossed the F1 generation. So he self crossed the F1 generation. So he has taken a P plant, right, uh, and within the flower, he transferred the pollen to the stigma. So now, so that is capital T, small t, and capital T, small t. So that is, these two will be the parents. Okay, 2n. Okay, of course, this is a heterozygous condition. Now, during the formation of gametes, so meiosis, so here this is a different allele and this is a different allele. So they pass on to different gametes. So here, or into one gamete capital T, into another gamete small t. So here also same, into one gamete capital T, into another gamete small t. So this is how the gamete formation takes place, that is haploid, right? So now, fertilization, so random. So here it is, we are going to see all the probabilities. So this one, if this one comes in, then capital T, then this plant will grow tall. And if this one and this one comes in contact, capital T, small t, so this is also becomes tall. And this one and this one, capital T, small t, so this also becomes tall. And this and this, small t, so this will be the dwarf. This is F2 generation. Now, after crossing the F1 generation, again Mendel obtained some plants. Okay, so suppose some 500 seeds he obtained. He sown the 500 seeds. Now when he sown all the 500 seeds, what he observed is that 3/4 of those plants were tall. So 3/4 of the plants were tall, and 1/4 of those out of 500 were dwarf. So the dwarf character, which was not expressed in F1 generation, was expressed in F2 generation. So with this, he came to know that the tall character is a dominant character and dwarf is recessive. So in the F1 generation, the character that is expressed, so he has taken it as dominant, and the one which was not expressed in the F1 generation is recessive. Okay, the character which was not expressed in the F1 generation was expressed in F2 generation. So that's what, when he saw all the plants, 3/4 of the plants were tall and 1/4 of the plants were dwarf, right? Fine. So that's about the Mendel's monohybrid cross, or inheritance of one gene. So it is inheritance of one gene. Okay, now here putting these lines in this way is cumbersome, and it leads to confusion that which gamete is fusing with which one. So hence, to avoid this confusion, and another person named Reginald C. Punnett, Reginald C. Punnett. So this person have given, right, something called Punnett Square. Okay, so to show the probability of all the gametes, okay, so he has proposed a Punnett Square, okay, Reginald C. Punnett. C. Punnett. So he is a British genetist, and he have given a method to show gamete probability in a easy way. So he showed gamete, okay, probability in a easy way, to show, okay, gamete probability in a easy way, of course without any confusion. So that's the Punnett square. So how I can define this Punnett square? So this is a graphical representation. So it is a graphical representation to calculate. So graphical representation to calculate the, okay, to calculate the probability of all possible, probability of all possible genes in a cross. Okay, possible, okay, of course, or possible genotypes. So possible genotypes of offspring in a cross. Okay, so this is how we define, is a graphical representation to calculate the probability of all possible genotypes of offspring in a cross. So that's the Punnett square. So we know the gametes that were obtained, okay, in in the second cross. So we'll take here the same monohybrid cross. So here I'm going to mention male gametes on one side and female gametes on one side. Okay, so that's what capital T, small t, capital T, small t I have obtained. Okay, so these are the four gametes which I have obtained. So these I'm going to put here. So one capital T and small t. And again, okay, so this is what, this is one parent. So one parents on one side, say this is acting as male and this is our female. Okay, so these two here. Now capital T, capital T. So capital T and small t. This one and this one, capital T, small t. Whereas small t, small t. So here this is tall, this is also tall, this is also tall, and this is dwarf. Okay, so you have to keep in mind that if one if one dominant character and one recessive character are present together, then it is the dominant character that will get expressed. So here dominant and recessive characters present together, then it is the dominant character that is, of course, the character that is expressed, we have to call it dominant, and the character that is not expressed is recessive. So that's what I'll just mention this. Okay, so here the character that is expressed in F1 generation, so that is dominant, and the character that is not expressed in F1 generation is recessive. Okay, so here in the monohybrid cross, when Mendel crossed the tall and the dwarf, what he obtained, all were tall. So there is no single plant that is dwarf. Okay, So and again when he self-crossed the F1 generation, in the F2 generation, okay, so this dwarf character was expressed. It means that in the F1 generation it was hiding, okay, and in the next generation it got expressed. So hence he concluded that tall is, okay, dominant, and dwarf is recessive, right? So this is what we call it, the Punnett square is called Punnett square, right? So that's about the inheritance of one gene. Now let's derive the phenotypic and genotypic ratios of monohybrid cross. So phenotypic and genotypic ratios of monohybrid cross. Okay, so here first we'll see about the phenotypic ratio. We'll say about phenotypic ratio. So coming to this phenotypic ratio, right? So here we have how many tall we obtain. So here we know 3/4 of the plants were tall and 1/4 were dwarf. Okay, so I'm going to take again the Punnett square. So male and female gametes, capital T, small t, and here capital T, small t. So capital T, capital T, capital T, small t, capital T, small t, and small t, small t. So here this is tall, this is tall, and dwarf. So you can see in this case there were 3 tall. Okay, so tall are 3, and coming to dwarf is only 1. So the phenotypic ratio is 3:1. And coming to the next one, the genotypic ratio. So this is a ratio of only monohybrid cross. Okay, the phenotypic ratio of monohybrid cross, and now let's see the genotypic ratio of monohybrid cross. So here in this case we need to take how many are homozygous tall and how many are heterozygous tall and how many are homozygous dwarf. So here in this case, in the Punnett square, you can see there is only one that is homozygous tall. So this is 1. Heterozygous tall, you can see capital T, small t, heterozygous, this is 1 and this is 2. So here heterozygous 2. And homozygous dwarf, this is 1. So hence the ratio will become 1:2:1. Right, so that's about phenotypic and genotypic ratios of monohybrid cross. So with this I will end today's session. In tomorrow's session we'll see the inheritance of 2 genes.